I think that's all. So again, I'm encouraging you to come to one of these social hours, some familiar faces. I promise you don't even have to talk if you don't want to. We have lots of people that like talking, me being one of them. And survey 7 away from 200. So fill that out if you haven't. And yeah, keep doing what you're doing. Everything's going great. Keep up the posts on Twitter and on Slack. So happy to see everybody. And now let's get to Carsten. This is going to be a good one. All right, there you go. Let's do it.
Perfect. Thanks so much, Kelly. Thanks for the nice introduction. Thank you for having me. And also thanks so much for organizing this Blood and Bone webinar, I think, which gives our day structure in these days and helps to keep all of us sane. So for those of you that don't know me, my name is Carsten Deppermann. And I did my PhD with Bernhard Nieswand in Würzburg, studying the role of platelet granules in thrombosis, hemostasis, stroke, and inflammation. After that, I moved on and went to Canada, to Calgary, to Paul Koop's lab. And there I learned intravital microscopy to study interactions of the innate immune system with bacteria and platelets. And my most favorite macrophage, the Kupffer cells. And this is where this story originated from. And we came back to Germany last year, and since December, I'm a junior PI at the University Hospital in Hamburg and trying to set up my own lab. But today I want to talk about this project, studying platelet clearance using intravital microscopy. The human body produces and removes 10 to the 11 platelets every single day. Platelets are produced by megakaryocytes, which reside in the bone marrow, which extend proplatelet protrusions into the vasculature, where they are shed into proplatelets and finally into platelets, which then enter circulation and circulate for 8 to 10 days in humans or up to 5 days in mice. At the end of the lifespan, platelets are cleared in the liver by a process that is yet incompletely understood. The first studies on platelet clearance date back to the 1960s, when Richard Astor and colleagues injected radioactively labeled platelets into healthy human subjects, and then using scintillation, finding that the majority of platelets are destroyed in the liver. Such kind of experiments, of course, are no longer allowed, but I hope by the end of my talk, I can convince you that, or I have convinced you that intravital imaging of the mouse liver is a great way to study platelet clearance. So what are we talking about when we talk about platelet clearance? There are different types of platelet clearance, the first being platelet in vivo aging. And this basically just describes the process of the platelet as it circulates in the human bloodstream, getting older and losing its functionality, and at some point needs to be removed from circulation. The second one is platelet in vitro aging, which happens, for example, during cold storage. You probably all know that if you store platelets in the fridge and then transfuse them into humans, they get rapidly cleared from circulation. So most of these platelets don't even really make it to the circulation. And because of this, platelets have to be stored at room temperature, which significantly limits their lifespan due to bacterial contamination. And thirdly, platelet antibody-mediated clearance, such as taking place during ITP, and these are mostly mediated by antibodies against platelet receptors GP1B or GP2B3A. So according to the current theory of platelet clearance during in vivo aging, platelets use the sialic acid, which are present on their surface. And this is depicted here with this purple diamond shape, and thereby exposing a galactose residue. This is this yellow circle here, which is then recognized by the Ashwell Morell receptor on hepatocytes. And they then take up the platelets and phagocytose them and destroy them. And this in turn turns on the JAK-STAT pathway, which leads to T4 production by the hepatocytes. And in contrast to this, in vitro aging, for example, during cold storage, And they not only lose the sialic acid, but also the galactose, and this exposes a glucanac residue, which is then able to interact with integrin alpha M beta 2 on macrophages. So, and now I would like to show you some of the studies that led to the establishment of this of this theory, basically. And then after that, I would like to show you some of our own data. So this is data published in Karen Hofmeister's seminal paper in 2003, where they showed that if you inject room temperature stored platelets into mice, they gradually are cleared and you have a half-life of about 36 hours. And then in contrast to this, when you have cold stored platelets, and this is the very bottom line, you see how rapidly these are cleared from circulation just within the first 1 or 2 hours. And interestingly, also this could not be rescued by adding cytochalasin B or EGTA. So calcium chelating or actin filament capping did not have an impact here. And so these processes apparently are not involved in this process. Interestingly, then, when they compared platelet aggregation capacity between the room temperature stored and the cold stored platelets, they found that there was no difference between the two. They also investigated where these transfused platelets end up, and this graph for room temperature platelets shows that most of them end up in the liver or spleen. And when they then look at cold stored transfused platelets, you see that the majority of them ends up in the liver. And they performed some sectioning and imaging on the mouse liver and stained for the platelets that were transfused with CMFDA, or for the macrophages using microspheres. These are stained in Nile red. And the overlay between green and red gives a nice orange. And as you can see in the merge picture here on the left-hand side, basically all of those platelets ended up in macrophages. And again, here they infused cold stored platelets with a TRITS dye, and this is an image from the liver. And you can see that there are significantly more of those platelets adhering than compared to when you transfuse room temperature platelets. And they actually quantified this. And this is this bar here in a wild type mouse. You have 4 times more cold stored platelets accumulating in the liver as compared to room temperature stored platelets. And interestingly, when they then used a CR3 knockout, and CR3, that's MAC1 or CD11B, CD18, alpha M beta 2 integrin, this was basically down to 1, indicating that this receptor is responsible to mediate the cold storage clearance. They then went on and checked what would be the co-receptor or the counter-receptor for MAC1, what would be the counter-receptor on the platelets, and they looked at GP1B, and this is electron microscopy using immunogold labeling for the GP1B receptor. And on a room temperature stored platelet, you can find that there is very even distribution of this receptor. And in contrast, on the right-hand side, you see a picture from a cold stored platelet, and you see how the GP1B forms these very big clusters of GP1B on the surface. So they, from that, deduced that GP1B might actually play a role. And indeed, when they then used an endopeptidase to cut off the extracellular domain of the GP1B, they could show that even room temperature stored platelets showed a significantly better survival. And when they looked at cold stored platelets, there was their survival improved even further and almost looking like normal platelets, indicating that cold stored platelet clearance is definitely GP1B dependent. In another seminal paper, they then followed the question if they could block this interaction of platelets with the receptor that mediates its clearance. And because it's known that MAC1 has a lectin domain, they looked at monosaccharides to compete with this interaction. And what they found is when they used beta-glucnac, basically competing for the binding of the glucnac to the MAC1, they found that already with low concentrations there was significantly less phagocytosis. And this is an in vitro assay where they incubate the platelets with the THP1 cell. That's a monocytic cell line here. So they thought, okay, well, that's a good idea. How about if we then cover these glucnac residues with galactose? And that's what they did here. And there's lots of lines in this figure. I'll just take you through it. So we have, again, it's survival of these platelets over time. And we have the room temperature stored platelets in the middle here. This is this line with the half-life of about 36 hours again. Then we have the cold stored platelets. This is the very bottom line, again, showing a very, very rapid drop in platelet counts. Now, they then incubated the cold stored platelets with UDP galactose as a substrate and this galactosyl transferase, which then is the enzyme that actually does the transfer of the galactose on top of this glucnac. And what they could see is that they survive significantly better, even better than the room temperature stored platelets. And what was even more interesting was that they didn't even need the galactosyl transferase for that. So just incubating these cold stored platelets with the UDP gal as a substrate was enough to significantly improve their survival. And they said this is because of the endogenous galactosyl transferase activity that is there on the platelets. So their conclusion from this paper was that the simple addition of UDP galactose to platelets, to these platelets bags, could offer a compelling strategy to secure the circulation of chilled platelets. And of course, the big question here is if this also holds true in humans. And indeed, there was a follow-up study in 2008. And to cut a long story short, well, it didn't improve the recovery of the platelets when they added UDP galactose to these to the platelet concentrates and gave them to these subjects. So this is the UDP galactose treated ones, and then we have the untreated ones. So there was no difference between the two. So going back to the drawing board, and now looking at different sugars, this time looking at the sialic acid, which is basically the terminal molecule here on top of the sugar chain. And this study in 2008 looked at ST3-gal4, which is a sialyl transferase, which transfers the sialic acid on top of this sugar chain. And the ST3-gal4 is of big importance for platelet sialylation. And you can see this here. Again, this is platelet survival, and in wild type or in heterozygous mice, you see curves that look very much the same to what we have seen before. But then in the knockout, you see just how short-lived these platelets are. They are basically cleared in a very, very short time frame. And this is presumably because they have an endogenous defect in sialation. So they are endogenously desialated, so to say. And they also did some tissue sections and stainings in these mice. And on the right-hand side here, you can see the ST3-gal4 knockouts. And this is a liver section where they stained for Kupffer cells using an F480 antibody here in red. And you can see that the platelets in green, they're all binding to these Kupffer cells. And below that, you see hepatocytes stained with albumin, and you can see how these platelets are inside the hepatocytes. And finally, they also looked at the spleen. And again, using F480 to stain for macrophages, they also found platelets in these splenic macrophages. Then they went on and further investigated the uptake of the platelets by hepatocytes. And here they used HepG2 cells, which is a cell line. And in this in vitro assay, they incubate the HepG2 cells with CM orange labeled platelets. And then they basically quantify the amount of HepG2 cells that take up the CM orange. And they used asialofetuin, which is able to block the Ashwell Morell receptor. And they could show that asialofetuin dose-dependently decreases the uptake of these platelets by hepatocytes, indicating that it might be mediated by the Ashwell Morell receptor. And then they also wanted to quantify what the relative contribution of macrophages and hepatocytes are in this process by depleting the macrophages. So if you look at this lower line, here they transfused ST3-gal4 knockout platelets into a wild-type mouse, and you can see this rapid clearance. And in the center here, they transfused ST3-gal4 deficient platelets into mice that had been depleted of macrophages using clotronate, and you can see there is a significant improvement in the platelet survival. However, there's still a big gap to the wild-type situation here, where wild-type platelets were transfused into wild-type mice. And they concluded that it's both hepatocytes and macrophages playing a role in this process. So coming to one of the more recent publications, and this is Renata Grozowski's paper in Nature Medicine 2015, where they looked at the Ashwell Morell receptor knockout mice and ST3-gal4 knockout mice and checked for their platelet parameters. And starting with the ST3-gal4 knockout mice, you see that they have significantly lower platelet counts, shown here compared to wild type. Also, their platelet half-life is significantly reduced, which makes sense because these platelets are just constantly cleared from the circulation, and they have a huge immature platelet fraction, indicating that there's a lots of young platelets, indicating that there's a lot of platelet production going on all the time. And also the RCA signal was very strong, indicating that these platelets all expose high numbers of galactose, and that's because they're endogenously desilated. And then going to the Ashwell Morell knockout mice, their platelet count is significantly increased. The platelet half-life is increased, and indicating that these platelets circulate longer. And as a consequence, the immature platelet fraction is smaller because there's less need for platelet production. And galactose levels in these platelets are increased, indicating that we have partially decylated platelets circulating that don't get cleared in these mice. And then, of course, the obvious thing to do was to cross these mice together and look at the platelet parameters in the double knockout. And they were able to normalize platelet count, somehow normalize platelet half-life and immature platelet fraction. However, of course, the galactose exposure still remained high, indicating that here these heavily decylated platelets continue to circulate. And another interesting finding was that in the ST3-gal4 knockouts, there was much more TPO in the liver, indicating that there was more need for the platelet production. And the opposite was true for the Ashwell Morell receptor deficient mice. And lastly, looking at this figure, again, we have lots of lines here. Now let's just focus on the green ones. So the lower green, for example, is here they transfused ST3-gal4 knockout platelets into wild-type mice, and as shown before, they are rapidly cleared from circulation. But then when they transfused these ST3-gal4 knockout mice into the Ashwell Morell receptor knockout mice, their survival was significantly improved. Finally, they also again use this HepG2 assay where they have the HepG2 cell lines and incubate them with decylated platelets, which are labeled with CM orange, and then they look for the CM orange positive hepatocytes as a measure of phagocytosis. And they could show that when they incubated them with decylated platelets, there was significantly more CM orange positive HepG2 cells. They also looked for TPO production by the HepG2 cells, and they could show that here TPOs is stained in red. When they added decylated platelets, the hepatocytes produced significantly more TPO, and this could be downregulated using Ashwell Morell receptor siRNA to knock down the Ashwell Morell receptor, or by using this JAK-STAT inhibitor. So, and from all this data, this all came together in this current theory of platelet clearance, that platelets, while they circulate, they lose the sialic acid, and then the galactose, which is exposed, is recognized by the Ashwell Morell receptor and then taken up by hepatocytes. And during ex vivo aging, both sialic acid and galactose are lost, and then the glucnag is recognized by alpha M beta 2, and these platelets get cleared in macrophages. However, there's a number of open questions that remain with this theory. And that was the reason for us to start this project, where we aimed at delineating the cellular players in platelet clearance. And what we did was we followed the fate of decylated platelets in vivo using intravital microscopy of the mouse liver. This is our setup. We use a spinning disc confocal microscope, where we have a heated stage, where we put our anesthetized mouse. We carefully expose one of the liver lobes and place it on that heated stage. The tail of these mice is cannulated, and we can then inject additional anesthesia or antibodies during the imaging. And using this technique and imaging the liver, this is what we usually see. We have here the endothelium stained in green. We have the Kupffer cells stained with an FR80 antibody in purple. These are intravascular macrophages. And then we have the hepatocytes in green. And the beauty of this is that we have all of the potential players that could play a role in platelet clearance, that we are able to visualize them and follow the processes going on live, basically. Yeah, and using a CD41 YFP reporter mouse, where the platelets are labeled with this YFP dye and they appear green. And looking at the liver, this is what we see. You see most of these platelets just zooming by. However, there's a small fraction of these platelets that get stuck to Kupffer cells, and the Kupffer cells again are here in blue. And you can see how they are probing their surrounding for what's going on. And there's a small fraction of the platelets that get stuck to the Kupffer cells, and you can even see this guy here trying to escape, but the Kupffer cell doesn't let go of him. So it looked like there was something going on with the platelets and the Kupffer cells. And this is, again, some still images to show you that lots of the platelets form really short-term interactions with the Kupffer cells. However, for example, this guy here, he is stuck on the Kupffer cell and stays there for 20 minutes and longer. And here on the right-hand side, you see a graph where we quantified more than 1,700 interactions of platelets with 124 Kupffer cells. And you can see the vast majority of these interactions has a dwell time of below 10 minutes. But then you can also appreciate that there is a small fraction which really forms long-term interactions with the Kupffer cells. And we quantified this, and it's about 0.85 firm interactions per Kupffer cell. And if you then multiply this by the total number of Kupffer cells in a mouse liver, then you get pretty close to what the daily platelet turnover in a mouse is. So and then we were wondering, so what happens to these platelets once they have bound to the Kupffer cell? And this is a 3D reconstruction from a single Kupffer cell, which we obtained using sectioning of the liver. So we had a Z-stack, and then we did 3D reconstruction, and we find this YFP remnants inside the Kupffer cell, indicating that these platelets are indeed taken up by the Kupffer cells. So to study this further, we made use of a model which induces rapid platelet clearance, and this is injecting sialidase. So by injecting sialidase, of course, we remove the sialic acids from all of the platelets at once, and this leads to rapid clearance of the platelets. And this has been used previously and shown that then the platelet counts drop really fast. So when we inject sialidase into our mice and then do the intravital imaging, this is what we see. We have the Kupffer cells here in purple. They're sitting in the blue vasculature, and then you have the platelets coming up in white. And as you can appreciate, just minutes after injecting these platelets, injecting this enzyme, you see how these platelets accumulate and cluster on top of these Kupffer cells. And this is the quantification for that. And then using a powerful software tool called Imaris software, we did some analysis of the co-localization of platelets with different cell types. And we found that the vast majority of platelets co-localizes with Kupffer cells. A small percentage of them also binds to endothelial cells, and basically none of them we found associated with hepatocytes. To exclude any potential side effects of the systemic sialidase treatment, we also used ex vivo desalation, where we bleed our mice to obtain heparinized whole blood. We then do a few washing steps to generate washed platelets. We treat them with sialidase and can then label these desalated platelets with cell tracker green dye. And we confirm that they have been desalated. And we also have a control group, which we leave untreated and then label them with a cell tracker red dye. And then we mix these two in a 1-to-1 ratio and inject this mixture into our mice and perform intravital imaging. And what we see is this. On the left-hand side, we have a control mouse, and we see much more green platelets, which are the desalated platelets, getting stuck to the Kupffer cells. And in contrast, we have the red platelets, which are the control platelets, and most of them continue to circulate here. And on the right-hand side, we have a clodronate-treated mouse, which means that most of the Kupffer cells are gone in this mouse. And both the desalated and the control platelets both continue to circulate. And this is the quantification for that. We also used another technique to measure the platelet count using flow cytometry. And we found that 24 hours after sialidase treatment, platelet counts are basically down to less than 5%. And then when we pre-treat these mice with clodronate to deplete the Kupffer cells and then inject sialidase, we found that we can rescue this drop in platelet counts and that they have about 80% of the normal platelet count. And we also measured the galactose exposure on these circulating platelets, and we found that there's a huge increase with the RCA fit signal, indicating that it is indeed desialated platelets that are circulating in these mice. And the next step, we also wanted to know if endogenous desilation would lead to a similar effect, and we used the ST3-Gal4 knockout mice. And as you can appreciate from this image, what you see here, actually, what you should see, we have the Kupffer cells in blue, but you basically can't really see them because they are all clustered by the platelets, and the platelets are in red. So all of these Kupffer cells are heavily clustered with these platelets, and there's only very few platelets actually circulating. And it looks like all the platelets, once they have been generated, they immediately get stuck on one of these Kupffer cells. And we can also nicely see this when we look at the platelet count under normal conditions in these mice. They are like less than 5% of a control mouse. But then if we deplete the mice of Kupffer cells, we can basically restore platelet counts up to 80%, indicating that it is indeed the Kupffer cells that play the important role here. So we then asked ourselves, okay, so what happens if we deplete the Kupffer cells? So what would happen to the platelet, the overall platelet population? And so we gave the mice a single clodronate injection and then followed the platelet population over time. And we have here the galactose on the y-axis and the CD41 on the x-axis. And as you can see, this population on day 5 shifts up to higher galactose exposure. And then on day 10 starts to shift down again. And this is also the quantification. You can see this nice peak here around day 3, 4, 5. And then on day 10, it starts going down again. And interestingly, day 10, that's about the time when you see significant amounts of Kupffer cells coming back into the liver after clodronate depletion. So these two really coincide with each other. We also looked at the young platelet population, and we stained it using thiazole orange. And what you can see in the Kupffer cell depleted mice, that there is a shift of the whole population to lower thiazole orange values. And also in the quantification, you can see that there is significantly less young platelets in these Kupffer cell depleted mice, indicating that there might be a feedback loop to prevent or to slow down platelet production in these mice. And taken together, these two effects lead to a small but significant increase in the overall platelet count in these mice after Kupffer cell depletion. Next, we were interested in finding out if the decylated platelets, once they have bound to Kupffer cells, if they also get phagocytosed by the Kupffer cells. And for this, we labeled our decylated platelets with a cell tracker red dye. And you can see these platelets have bound to the Kupffer cell. And then over time, they start releasing this dye into the Kupffer cell, thereby actually staining the body of the Kupffer cell red, indicating that indeed these platelets are being phagocytosed and then broken down to release this dye into the cell body. And this is also a 3D reconstruction of this, where you can see that large parts of the cell body are actually stained with this cell tracker dye. Next, we wanted to address the question if also the spleen might be involved in this process. And to this end, we splenectomized our mice and then treated them with salidase. And we have on the left-hand side, we have control platelets before and after 24 hours after salidase treatment. And you can see this big drop. And then on the right-hand side, this gray bars, this are splenectomized mice. And you can see that they're basically after 24 hours, the platelet counts are pretty much the same compared to the control mice, indicating that spleen doesn't seem to play a major role in this process. We were also wondering if these decylated platelets get stuck in the lung, and for this we used intravital imaging of the mouse lung and injected salidase into these mice. And here we have the endothelium stained in red and platelets stained in green. And as you can see from these images, but also from the quantification over time, there's no increase in platelet signal here. So there's no platelet accumulation in the lung after salidase treatment. So we were then wondering what is the receptor that mediates this platelet clearance? Is it the Ashwell Morell receptor? And most of the time, the Ashwell Morell receptor is only described as a hepatocyte receptor. However, there are also reports that say or that show that it is expressed on Kupffer cells. And asialofitoin has previously been used to block the Ashwell Morell receptor. So we pre-treated our mice with the asialofitoin on the right-hand side here, and then injected salidase. And you can see that there was significantly less platelets adhering in these mice compared to our control mice, where Kupffer cells were rapidly clustered with platelets. And this is the quantification. So we were then wondering, is it really the Ashwell Morell receptor? And to really prove the point, we used the Ashwell Morell receptor knockout mice. And to our surprise, after salidase injection, we found there was no difference in the accumulation of platelets on the Kupffer cells in these mice. So we went back to the literature and checked if there are other known ligands for the asialofitoin. And indeed, it is known to be a ligand for the macrophage galactose type lectin, which is MGL. And what was interesting that just a couple of years ago, 2017, there was a paper showing that the macrophage galactose lectin is actually also involved in the clearance of von Willebrand factor. So this got us interested, and we went to buy the MGL blocking antibody, injected this one into our mice, and again injected salidase to induce platelet clearance. And to our disappointment, there was no difference in the accumulation of platelets and the MGL blocked mice. However, when we then combined MGL blockade with the Ashwell Morell receptor knockout mice, we found that there was significantly less platelets accumulating in the livers of these mice, and this is shown here with this orange bar, indicating that it is a combined effort of the Ashwell Morell receptor and the MGL to then mediate the clearance of these platelets. Next, we also were interested if in the mice with the aged platelet population, so to say, after Kupffer cell depletion, if this has an impact on the hemostatic capacity of these mice. And to check this, we use this bleeding time assay, where we cut the tail tip, the last millimeter of the tail tip of the mice, and then we check how long it takes for the bleeding to stop. And in a wild-type mouse, this usually doesn't take much longer than 5 minutes. But we were surprised to find that when we used our Kupffer cell depleted mice on day 5, when we found this peak of galactose exposure in this population, that none of them actually were able to really stop the bleeding, indicating that they have a significant hemostatic defect. And we saw very similar results when we looked at the amount of blood loss. So they significantly lost more blood. And finally, we also asked ourselves if this process would also be involved in the clearance of cold stored platelets. And we injected cold stored platelets here labeled with a cell tracker dye, and we found them accumulating on Kupffer cells in the liver of our mice. And in the next step, we also wanted to check if the Ashwell Morell receptor and MGL are also involved in mediating this clearance. And indeed, while the Ashwell Morell receptor alone didn't have any impact on the clearance, when we combined Ashwell Morell receptor deficiency with the MGL blockade, we found that there was significantly less platelets accumulating in the livers. And with this, I would like to summarize. We have now found that aged or cold stored decylated platelets expose galactose residues, which are most likely recognized by MGL and Ashwell Morell receptor combined on Kupffer cells, and that this leads to the uptake and destruction and clearance of these platelets from circulation. So when we deplete the mice of Kupffer cells, this leads to an increase in the number of old platelets, a decrease in the number of young platelets, and taken together leads to a rise in the number of total platelets. And this shows that Kupffer cells are crucial to maintain a healthy platelet population. And with this, I would like to thank my former supervisor, Paul Coops from the University of Calgary, all members of the Coops lab. I would like to thank Jamie Marth from the University of California, Santa Barbara, for providing us with the Ashwell Morell receptor deficient and the ST3-Gal4 mice. I would like to thank the DFG for funding. And while I have your attention, I would like to ask you to save the date for the Platelet and Megakaryocyte Gordon Conference, Gordon Research Seminar taking place February 2021 in Tuscany. And the guys from the GRC and also us from the GRS, we're trying to put together a really, really great program. And this will be an excellent opportunity for trainees, but also PIs for networking and sharing some great science. And if you're interested in working with us, please drop me an email. And yeah, thank you so much for your attention, and thank you so much. And I'm happy to take questions.
So good. I love the story so much. We already have some questions, so why don't you stop share?
Yes. Okay. Thank you. Okay.
I just feel like such a complicated thing, and so many people have contributed lots of pieces to the platelet clearance story. And I think your work really underscores the complexity and that like these pieces can fit together in different ways. So
yeah, it's definitely a complex story, and there's definitely a couple of lots of parts of puzzles that have to fit together.
Definitely.
Let me just quickly have an.
It's okay. Don't feel overwhelmed. You can go one at a time. Good to get questions.
Yeah.
If you want, I could read that
Yes.
to you.
So, did we? Alex is asking if we try platelet aggregation with old platelets to compare with our bleeding time in vivo. Yeah, and yes, we did that. We did platelet aggregation, and they were defective. So what we found was that they are they do form an aggregate, but it seems to be less stable. So all the agonists we tried, they formed an aggregate, but then it dissolved again. So it was more of a kind of a transient aggregation taking place there. So Raja Ward is asking, just wondering if you have insights as to how mechanistically MGL and ASGPR may be working together. Yeah, I would love to know that. Right now we don't really know. We are, well, at least I am convinced that it's the receptors on the Kupffer cells, because some people say it could still be the hepatocytes, but then the hepatocytes extend these protrusions through the fenestrations of the liver endothelium into the vasculature. But we have used a method which gets rid of these fenestrations. This is if you give the mice really a highly diluted arsenic dilution to drink. It's like 250 parts per billion, so highly diluted. But this has been shown to reduce the number of fenestrations of this liver endothelium. And we haven't seen a difference in both the clearance and also the platelet count in these mice, indicating that yeah, it's not an interaction taking place on these hepatocytes protrusions.
Is this a question you're working on now? Is this where your work is going?
I am, well, as I said, I'm currently just establishing my lab and waiting for the nice intravital microscope to arrive at some point, hopefully. So yeah, working on this. Actually, I would also be interested in studying what's, I don't know if someone asked about this, but to look how this feedback really works to the platelet production with the megakaryocytes. If it's only just via TPO, or if there is more to it, right? If there's a closer
yeah. So Jamie, hi Jamie, asked, do you see any TPO production in platelets cleared by Kupffer cells, given that the Ashwell Morell would be
I would have to. Unfortunately, we never did this TPO quantification in the plasma of these mice. We should have done it. Yeah, unfortunately, we never did it. Yeah, but that would be really interesting, and that would be basically the starting point for that question, right?
Wally asked if the decylated platelets have any effects on the Kupffer cells.
Yes, interesting question. Especially in these, if you look at these ST3-gal4 mice, right? They have constantly, so these Kupffer cells, they are just constantly eating the platelets, and you wonder, well, this affect their capability to do other things, right?
Yeah.
For example, I mean, we have shown, or Paul has shown that the liver Kupffer cells are really important in clearing bacteria from the bloodstream, for example. So would these mice have a defect then in clearing bacteria? So that would be an interesting question, yeah.
Or maybe vice versa. I don't know. I'm just wondering about like bacterial, like sepsis and platelet counts, if there could be some effect.
Yeah, I mean, what they have done also is that they found that when the Kupffer cells, when the bacteria are caught by the Kupffer cells, then the platelets also start to accumulate, and then they build this kind of aggregate around it to somehow encapsulate the bacteria on top of the Kupffer cell.
That's crazy.
So if you have already a lot of platelets there, yeah, does it help or does it make it worse? I don't know.
That's really crazy. Ren Howe has a couple questions.
Yes, let me see.
If the exposed galactose is recognized by the liver, can you speculate why the red blood cells are not cleared after neuraminidase injection? Observe if red blood cells are attached to the Kupffer cells.
Yeah, that's an interesting question. We have never really looked at red blood cells, but what I know, and Paul actually did some studies back in the day. And what is interesting about this is really mouse specific. So in the mice, this sialidase does not affect the clearance of the red blood cells. But if you look at in humans, for example, it does. So if you would do the same to a human, they would get cleared. And it's also true for cats and monkeys, I think. So there is definitely something to it, but it doesn't happen in mice, which is interesting. Yeah, but we haven't really looked at that. I'm sorry.
We also asked if there's evidence that MGL and AMR are physically associated.
Physically associated. I don't know. No, I don't really know anything about that. What I would love to do, because I mean, both Ashwell Morell receptor and MGL, they both come in two. So there's ASGR1 and 2, and there's MGL1 and 2, and they basically both sit together in the same cassette on the chromosome. And what I would like to do, just to knock out all 4 of them. Of course, you can only do that via CRISPR, right? You didn't want to breed that. We tried. We started breeding some of those, but of course, this is an insane thing to do. You'd have to do it via CRISPR. So looking at these questions, yeah, I would love to do that.
Definitely need more people in the lab.
Definitely.
Daphna has a question. Have you looked at platelet von Willebrand factor complexes and clearance? Is the sialidase treatment having an effect on that as well?
Yeah, interesting question. We haven't looked at von Willebrand factor, but we have looked at GP1B in the GP1B deficient mice, and actually they show less clearance actually. So treating them with a sialidase has less of an effect actually, yeah. Which makes sense because there's literature that says that most of the sialic acid is on the GP1B.
Okay. Yeah.
Yeah.
That makes sense. So Jerry's asking about differences between human and mice platelets that could account for the different response to UDP, gal addition, and cold storage platelet clearance. Oof, lots of differences, right?
Ooh, yeah, lots of differences. Yeah, I assume. Yeah, of course. Yeah, that might be the reason. Might be one of the reasons there. But we unfortunately never looked at human platelets. We only did this in mice. No.
Ooh, we have a question from Abs, which sounds like he's trying to make you solve their problem. Let's give it a chance. He says they have a mouse model which has a really pronounced microerythrocytosis and platelet IPF. Mice have large splenomegaly. Should we be looking at the liver? So let's make this more generalized. Have a weird phenotype. Are we missing things by not looking at the liver with platelet clearance?
Yes. Well, if you ask me, it's always worth looking at the liver. But well, if you have a splenomegaly, if you have a huge spleen, that tells you that there is something going on in the spleen, right? But you could also be looking in the spleen. You can do intravital imaging of the spleen too. It's basically as simple or as difficult as the liver prep. You can easily do that.
I mean, this is kind of like a dogma. I don't know if it's true, but I've always been told the spleen is just so different. Like the function of the extramedullary hematopoiesis in the spleen in mice versus humans is so different that it's a lot of times just not looked at as relevant in mice because it oftentimes doesn't really.
It doesn't do the same things as in the human, yeah.
Because there's a ton of megakaryocytes in the spleen in mice, but no one's really seen the same in humans.
Yeah.
So,
yeah, it is interesting.
Not sure. Just putting that out there.
Yeah.
Sorry, Abs. I mean, that's what Carsten's here for. He's your expert, so ask away. And he gave us time for questions. So thank you. I think that was a lot of questions. So thank you all. And thank you for sharing this work with us.
Thank you so much.
So since we have a couple more minutes and you're looking for people, you hit on this a little. Do you want to give a little plug for what you are looking for? What projects and things people might be interested in?
Oh yeah, no, that's a nice question. Thank you. One of the directions that we are going right now is still working on the liver and interactions between platelets and the innate immune system. Since there is also from the clinical side, there's a big interest in liver research here in Hamburg, and one of the specialties here is they are looking at PSC patients, so primary sclerosing cholangitis, which have these fibrosis of the bile ducts, and it's an autoimmune disease that no one really understands how the pathophysiology mechanisms are. And so there are a couple of mouse models to mimic this disease, and we were wondering if platelets would also play a role in that, because people have shown that there is something wrong with coagulation in platelets in these patients. And the cool thing is, because they have in the clinic, they have basically a group of PSC patients which they see regularly. So they get blood and also sometimes liver biopsies, or when they have to transplant the liver, they get the whole chunk of liver. So there's lots of human blood and tissue which we can analyze and then compare this to the mouse work that we have done, that we will be doing. And and in the long term or not so long term, we will also be doing then intravital imaging of the liver and also the intestine of these mice, because usually there's with PSC there's usually also it comes with inflammatory bowel diseases. So there's something going on between the liver and the intestine in these patients, and we will try to mimic this in the mice and then see if we can find any correlation.
So everyone go work for Carsten. That's like I really like the alternate functions of platelets. Like, it's something I think about a lot too, like megakaryocytes, because there are a lot of people in the platelet field, but there are some things like everything you just described that are pretty wide open spaces. So there's a lot of room there to like make your mark and do something cool and new. So cool. Well, definitely. I know you'll do great, so I don't have to wish you good luck, but
thanks so much, Kelly.
It'll be exciting to see where your lab goes.
Thank you.
All right, everyone, another great day. Thank you for joining us, and we'll be back tomorrow. So enjoy the rest of your Tuesday. Bye, everybody.
Bye.
Kristen.